Ladies and gentlemen, welcome to the Global Young Scientists Summit 2022. Today, I have great pleasure to introduce to you Professor Barry Marshall, Nobel laureate 2005. Professor James Barry Marshall is clinical professor and branded ambassador at the University of Western Australia. Professor Marshall and Emeritus Professor Robin Warren were awarded the 2005 Nobel Prize for Physiology or Medicine for their discovery of the bacterium Helicobacter pylori and its role in gastritis and peptic ulcer disease. Professor Marshall was at the Health Science Center at the University of Virginia for 10 years before returning to Perth, Western Australia, to his post as clinical professor of medicine at the University of Western Australia. Today, Professor Marshall is director of the Marshall Center for Infectious Disease Research and Training. This center is obviously founded in his name. And in addition to Helicobacter pylori research, the Marshall Center is also at the forefront of infectious disease identification, surveillance, diagnostics, and drug design, and transformative discoveries. His research has expanded with his group to embrace new technology, including next-generation sequencing and genomic analysis. As a personal friend of Professor Marshall, I know that he is a very nice person and also very innovative person. You will hear from him his amazing story of how he was driven by curiosity to discover this bacterium in the stomach, which was supposed to be unbelievable. And I'm sure you will enjoy his story, how he will be led to the path of getting the Nobel Prize. So today, his plenary topic is how curiosity-driven research resulted in the Nobel Prize in medicine. I give you Professor Barry James Marshall.
Hi there, I'm Barry Marshall. I'm Clinical Professor of Microbiology at the University of Western Australia in Perth. And it's my pleasure today to give you a lecture for the Singapore Global Young Scientists Forum. The title of my talk, as you see, is How Curiosity-Driven Research Resulted in the Nobel Prize in Medicine in 2005. So as you know, this discovery was related to the stomach bacteria Helicobacter pylori, and you'll learn more about that as we go through the lecture. Now, it's extremely common. I'm not going to talk about the epidemiology of it very much today, but we found out ultimately that about half the people in the world have this bacteria in their stomach, and it can lead to ulcers and, worryingly, stomach cancer. So we'll start off way back in 1979 when my colleague Dr. Warren, who's a pathologist, had his first case. And I think it was his 42nd birthday. And he's still got the records of that pathology report that he did. So this is what he saw. Now, initially, when you look at a biopsy, it's usually a H&E section. And so the tissue is pinkish, and the bacteria, if they are there at all, might just be very faint bluish smudges. So Warren saw these bluish things on the surface of the biopsy, and he wanted to have a clearer look, so he asked for a special stain called a silver stain, which stains the bacteria black and the tissues a golden color. And here you see his first case. Now the thing you'll see there is that you've got yellow cells, and these are the cells that line the stomach. The clear area is the mucus layer, and the black things there all over the place, especially on the surface of these epithelial cells in the stomach, are the bacteria. And we now know that they are Helicobacter pylori. But in those days, when we first named them, we called them Campylobacter. So Campylobacter means curved bacteria, and Helicobacter means a bit more, it's spiral, like a corkscrew. And Helicobacter pylori, the name we gave them in the end, means that a curved bacteria or a spiral bacteria living at the bottom end of the stomach near the pylorus. And pylorus is the Greek word for gatekeeper. And so that reflects the function of the pyloric muscle at the bottom end of the stomach. So that's how it got its name, and now we will just call it H. pylori, because Helicobacter is a pretty long name. So here's Dr. Warren's first slide, and you can see that all the curved bacteria stuck all over the surface of the epithelial cells, and they are black. And the magnification there is about 1,000 times. Now that's one of the reasons these bacteria had not been discovered before. They had been discovered a few times, but most pathologists didn't see them, because most pathologists will diagnose cancer, stomach conditions, etc. at a low power, say 250 times. And at that power, you cannot see the bacteria. So here's the curiosity-driven research thing. Bacteria are killed by acid if the pH is less than 3. So that's like strong lemon juice would be 3. Stomach has acid in it, which is pH of 1.5. So about 10 times or 20 times more acidic than the strongest lemon juice you can think of. So the question is, how can bacteria live in the stomach when there's acid everywhere? So that's why nobody ever looked for bacteria in the stomach, because common sense would dictate, oh, they can't live there because we know bacteria are killed by acid. So we wondered that. How can they live in the stomach if there's acid there? And we started this collaboration, and I started with Dr. Warren in 1981, and we worked pretty intensively on this project for about 5 years after that. And here we are after our first publication in The Lancet. And you know, The Lancet is one of the most widely read medical journals for clinical medicine in the world. And that was our first paper, really, or my first paper at least. And here I am in Dr. Warren's lab, and we're looking through his pathology books, noticing things about gastritis, stomach cancer, bacteria, etc. Warren and I did a study of 100 patients coming to have an endoscopy, which is the tube down the throat, look in the stomach for cancer or ulcers, etc. And so the fiber optic endoscope went down, and biopsies were taken for microbiology and pathology, and all the data was collected. And I studied it a few months later, and this is what I found. So in the bottom line, you see the total of 100 patients was that 58 or 58% had the bacteria in the stomach. So they were surprisingly common. However, when people had a duodenal ulcer, which is the most dangerous and most common form of a peptic ulcer, because you can bleed to death from a duodenal ulcer, you'll see that of 13 patients, all 13 or 100% had the bacteria. So that was highly significant. P-value is 0.00044. So it's like throwing heads 13 times in a row, or red 13 times in a row at the casino. And also gastric ulcers, which are ulcers in the stomach, 22 patients, 18 or 77% had the bacteria. So again, so highly significant. So we're pretty excited about that, and we wanted to follow it up and publish it. And this publication was in 1984. By that time, we had already been testing patients for nearly 3 years. So we were starting to understand that this was a consistent finding, and wherever we looked, we could find this association between the bacteria and ulcers. However, I ask you, would the world accept the new cause for ulcers on the basis of data from only 13 patients? No way, because it was an important discovery, and too important to suddenly change the paradigm. And the current paradigm at that time was that ulcers were caused by stress. And there were plenty of people that were making a living treating stress in ulcer patients. And now we know that stress has got absolutely nothing to do with ulcers. So no way. No one's going to believe it straight off. They want further evidence. And Warren and I tried to present this at the gastroenterology meeting in 1983. And we received a letter back from that conference planner. Dr. Marshall, I regret your research paper was not accepted for presentation. The number of abstracts we received continues to increase. And for this meeting, 67 were submitted. We could only accept 56. And so ours was in the bottom 20%. In fact, it was one of the worst ones as far as the judges were concerned. And they said, Dr. Marshall, I'm sorry, you haven't been selected to present your paper. Not only that, we're not even going to let you present a poster. It's a bit too strange. And we would be quite happy if you don't come to the conference, because it's a bit shameful to produce research like this. However, Warren and I were very confident that there was something going on, because we knew of previous discoveries of these bacteria in the stomach going back 100 years. And they had already been discovered, and recently I published a book called Helicobacter Pioneers, and you can see the table of contents here: 1892, 1917, 1940, 1950, 1957, 66, 73, 75, 78, 79, 73, 79, and 82 was Warren and Marshall. So about 10 other people had published done a thesis on bacteria in the stomach. Not always human, but sometimes dog stomach, mouse stomach, etc. So one of the most important one was this guy, Stone Friedberg, and he later became a famous professor at Harvard, and he was a cardiologist, I believe. But when he was a young man, he discovered that spiral bacteria lived in the stomach, and he had quite a few patients, and at post-mortem or at surgery of the stomach, they had samples showing that bacteria were present. This is him when I met him a few years later in Boston. And finally, this is when he was about 100 years old at Harvard Medical School. He's looking through his thesis there. So he discovered spirochetes in the human gastric mucosa and published it. And this is one of his pictures, a black and white illustration. But as you can see, the wiggly bacteria there inside a gastric gland look very similar to those ones that Warren and I were seeing in 1982. However, nobody believed it, because the top pathologist in the United States, Eddie Palmer at Walter Reed Hospital, he found that Friedberg and Barron were proven to be wrong, but unfortunately he had incompetent researchers working for him, and nobody knows how he missed it. But he said none of the 1,180 specimens was found to contain spirochetes or any structure which could reasonably be considered to be of a spirochetal nature. So he published this, he says, we looked at 1,000 cases and we didn't see any. So Friedberg's things must have just happened after you died, or after they cut the stomach out, other bacteria grew there. Nobody bothered about it. It was just useless information. And I'm sure Dr. Friedberg was a bit disappointed by that. Now, in the 60s, one person took an interest in them, Susumu Ito. He was also at Harvard, and he had one of the best electron microscopes in the world then, and he was describing the anatomy of the stomach, and he was looking in cat's stomach, and he saw spiral bacteria in the stomach of the cat. And so one day he said, well, I wonder if humans have these bacteria in their stomach. So he swallowed a biopsy tube, which is on the end of like a long cable, and on the end of the cable is a little knife which cuts a little piece of tissue out of the stomach, takes it out and stains it. So he had very, very fresh tissue, and he was very good at staining for bacteria, etc. And he produced this photograph, which is Helicobacter pylori, and it's actually from his own stomach. And we now know that Helicobacter pylori is extremely common in Asia, especially in Japan, where nearly 100% of people used to be infected with Helicobacter. And you know that was the #1 country for stomach cancer in the world until about 10 years ago. So here it is, Helicobacter, and you'll see on the end you've got a couple of flagella sticking out there, and it's firmly attached to the epithelial cell with all kinds of lectins and things there, which are not particularly well understood, but it's very, very interesting. So that was published in the Handbook of Physiology, and there he is, Susumu Ito at Harvard Medical School, when he and I were corresponding in 1995. But the question is, when you discover a new germ, how do you prove that it's a harmful pathogen? And there are a few rules about how you go about it. And one of the key things that you try to do is infect animals with the bacteria and see if it can produce some similar disease. in animals. So our experiments included mice, rats, rabbits, guinea pigs, pigs, and ultimately a human, which was me. So in preliminary studies, which were not well documented, we tried to infect the Helicobacter into mice, rats, rabbits, and guinea pigs, but no effect. So Helicobacter pylori is specially adapted for the human stomach, and it probably does not stick to animal stomachs very well. So therefore it can't cause the infection. And we tried to infect piglets, and I was feeding the bacteria to these piglets and doing endoscopies on them once a week to see if the bacteria had taken. But the pigs grew, and we found that after 6 months the endoscope was not long enough, because if you have studied pigs, you'll know they have a very, very long body, and so that the human endoscope wasn't adequate to do endoscopy on those animals. So the experiment failed. We could not infect pigs. So in 1984, a human volunteer was given H. pylori. Now, although it sounds pretty interesting to do an experiment on yourself, it's really not very scientific. And so I was always a bit embarrassed about telling people that I had drank the bacteria. So ultimately, the Abbott company, who made antibiotics against Helicobacter, you might have heard of it, clarithromycin was the best one, they produced a comic strip about my experiment where I drank the bacteria, and it was published in the Medical Journal of Australia, actually in 1985, not 1984, an attempt to fulfill Koch's postulates for Campylobacter pyloridis, and that's the name we had for it in those days. And here I am, my lab tech says you're crazy, and I'm there. There's no other way. And there wasn't any other way because we had tried it in animals. So the details of the experiment are that I underwent a baseline endoscopy, and looking in my stomach was completely normal. All the biopsies were normal, no bacteria, lots of acid. So I drank bacteria, which was 10 to the 9th colony-forming units, and that's how you measure bacteria. So that was 2 Petri dishes covered in growing Helicobacter pylori. So after 3 to 5 days, I felt vaguely unwell, and my mother and my wife both told me that I had halitosis, a bad breath. So in retrospect, the low acid level was allowing all kinds of filthy bacteria to start growing in my stomach and causing halitosis. I think that's my explanation of it. Then after day 6 to 10, I was vomiting, and the vomit didn't have any acid in it. So finally on day 10, I had another endoscopy to see if the bacteria had taken, and what do you know? I had severe gastritis. The bacteria were all over the place, and that proved that Helicobacter pylori was a pathogen causing this inflammation of the stomach, which we call gastritis. But I did not develop an ulcer, and you can read about the details of that experiment on Wikipedia if you want to. So here's my silver stain. The stain is a bit brownish on this occasion, a slightly different one, but you can see lots of those beautiful bacteria sticking to the epithelial cells. And if you looked at other parts of this biopsy, you could see lots of white cells in the stomach trying to eat the bacteria, trying to eradicate the infection. And I had an infection and was a bit unwell then for about another week. So that experiment was published in the Medical Journal of Australia in 1985, and people around the world were pointed to it by an editorial in The Lancet, and a bit curious about it and pretty interested. And one of the people that was interested was the microbiologist at Procter Gamble in Cincinnati in the USA. Now I know that Procter Gamble is a global company with all kinds of well-known brands. So for instance, soap powder, especially Tide and Bounty, different kinds of diapers, toothpaste, shampoo, you name it. But they also produced some gastric compounds for unsettled stomach, particularly in the United States, so that you would see why they were a bit interested in stomach treatments. And here I am at the University of Virginia in 1987, probably. So here's that medicine produced by Procter Gamble at the time, and that's called Pepto-Bismol, and had been used to treat people with gastritis or gastroenteritis, any kind of gastric upset, for about 100 years. But in its original formulation, 100 years before, it said in conjunction with other indicated remedies, it's been found of service in treating gastric ulcers as well as the relief of acute gastritis. So it had a bit of a history of being good for gastritis, even though there was not really good scientific data about this. So I had mentioned this in one of my papers, and that's what pricked up the ears of Procter Gamble. So bismuth, a close relative of arsenic, and arsenic was another thing that was used in small doses for all kinds of GI complaints before the era of antibiotics. And so it remains as a modern equivalent of Kobayashi's arseminol. So Kobayashi was the guy in Japan studying stomach bacteria, and he was treating them in mice with an arsenic compound called arseminol, which was also used to treat syphilis. The popular medicine Pepto-Bismol in the USA has been shown to kill Helicobacter and heal ulcers. So that was shown subsequently. And so Procter & Gamble, as you can imagine, were pretty excited because there were billions of dollars to be made by discovering new ulcer treatments. And that research goes on today. So here's an experiment I did in 1983, actually. And here you see a culture plate showing Helicobacter pylori. And in the middle, I've got a disc there, which has got some Pepto-Bismol, or bismuth subsalicylate is the chemical name of it. And you see a very sharp zone of demarcation, how the bismuth is toxic to the bacteria and they can't grow anywhere near the bismuth. And this is a picture of a biopsy, and this person took some Pepto-Bismol and 30 minutes later had an endoscopy and some biopsies. And the interesting thing is that these black metallic spots have precipitated all around the bacteria, so the bismuth gets concentrated by the bacteria and then kills them, and they detach from the mucosal cells. So really quick and really effective treatment, and maybe that's why it was so popular for stomach upsets for 100 years. Even today, people are talking about H. pylori and how you kill it. So usually you take an acid blocker and some antibiotics. And here's a drug company. This is AstraZeneca's mouse pad, which I'm actually using here today. So Losec HP7 shown here was the first really good treatment for Helicobacter, and that consisted of omeprazole, an acid blocker, and 2 antibiotics, which is amoxicillin and clarithromycin. So several drug companies teamed up to produce this medicine, and people have been taking it all over the world for 20 years. It's still one of the best. But in there, they were a bit worried that there was a lot of competition from bismuth, that Procter & Gamble treatment. And so they like to say no bismuth because some people reckon that bismuth was dangerous to take. It's not true. It's never ever caused any trouble. Even after 100 years, I've hardly ever heard of side effects from it. So it doesn't give you allergies like some antibiotics do. So that's one of the advantages. If you're allergic to penicillin, you can't take Losec HP7, you might take a bismuth treatment. So if you've got Helicobacter, don't worry, it can be cured. So bismuth and amoxicillin, one of the safest drug combinations with Losec. So Dr. Warren said I should do a double-blind study. So I did a placebo-controlled double-blind study in Australia in 1985. And what we did, we took patients who had Helicobacter, who had troublesome recurring ulcers, and we gave them 2 treatments. So they either got cimetidine and placebo. So one group got acid blockers plus fake antibiotics, and the other group got acid blocker, cimetidine, plus bismuth and metronidazole, both anti-Helicobacter treatments. And then we healed the ulcer, and then we waited about 12 months and from time to time did an endoscopy to see if the ulcer was coming back or if the patient had a complication of it. And what we found is that if the patient had received the bismuth with metronidazole and the Helicobacter had been eradicated, guess what? The cure rate was 80%. If you just received an acid blocker and no antibiotics, and you still had the Helicobacter in your stomach after that treatment, the cure rate was only 20%. In other words, 80% of people had their ulcer coming back after 12 months. So it was pretty obvious that this new treatment was really, really great. And subsequently, another study was done, this time in Austria, not Australia, and that was placebo-controlled, and that's similar, but ranitidine, that's another acid blocker, with amoxicillin and metronidazole, and compared with ranitidine and placebo. So if you look at this study, you see after the ulcers were healed, you just waited, and sure enough, all the patients who had received fake antibiotics had recurrence of their ulcers. So it went way, way down. So less than 10% of people were cured, compared with about 90% of people cured of their ulcer after the Helicobacter had been eradicated. So it was pretty obvious then, and people really believed that ulcers were now caused by the bacteria. So, hey, let's get rid of them. And guess what? Most people who developed stomach cancer, which is, as I said, is very common in Asia, most of those people had Helicobacter for most of their life, causing that inflammation, and we do think that inflammation leads to stomach cancer. So there were more double-blind trials after that. As you see, we studied 100 patients, etc. And then other people did it in Amsterdam in 1990, Dr. Graham in Houston in 1991, and then Henschel, as I showed you, in Vienna. So after that data, other people repeated it, as I said, and eventually there was a consensus conference in about 1994 at the National Institutes of Health in Washington, D.C. So after that, everybody could treat ulcer patients with antibiotics if they wanted to try it out. But the treatments weren't so good, and it was very difficult to see if the treatment worked because you had to pass the endoscope on the patient, take another biopsy. So that's like a $1,000 or $2,000 procedure. And so not everybody could afford it, and everybody was so busy they couldn't double the number of endoscopies that they were doing in their hospital. So it was a bit of a problem. So we needed some simple, accurate tests to diagnose Helicobacter. Firstly, to make it simple for gastroenterologists. You didn't have to have a research lab anymore. And then ultimately to make it simple for your regular family doctor to do the whole thing. And maybe you wouldn't even have to have an endoscopic examination at all. So that was the hope. So this is the answer to the curiosity-driven research idea that I showed you way, way back at the beginning of the lecture. So how can bacteria live in the stomach? So let me tell you about urease. It's an enzyme produced by Helicobacter. And it was discovered in Helicobacter by Dr. Langenberg in Holland in about 1986. The thing is, though, that of all the bacteria on the earth, Helicobacter produces more urease enzyme than anything else. So it must have a useful purpose, right? And I was looking at the equation there and I said, well, hang on a minute. Here's urea. It's present all throughout your body, in your gastric juice, your saliva and everywhere. So it's in your stomach. And it consists of CO2 with 2 ammonia molecules joined on. And if you have urease enzyme, for instance, from Helicobacter, that would be broken down. It would consume a hydrogen ion. So in the reaction, acid is used up. Takes up a bit of water and generates ammonia and bicarbonate. So you see, we've got acid on this side and we've got alkaline on the products. And so this is a way that Helicobacter protects itself from acid. And so that the curiosity we had, how could bacteria live in the stomach? What do you know? This is the reason. It makes ammonia and bicarbonate and protects itself from acid. So we did further experiments to prove that. And here you see an XY plot. On the x-axis, you see the acidity increasing as we go to the right, which is a lower pH down to 1.5. And we put different bacteria in this brew, which we called artificial gastric juice. And if the pH was 7, for instance, neutral, all the bacteria survived for about 30 minutes, probably for hours. And so the ones we tested was C. jejuni, no urease, Proteus, a little bit of urease, and H. pylori, lots of urease. Anyway, so if you look at what happened, you'll see This one, C. jejuni, after pH 4, as it gets more acidic, the bacteria are killed. So there's about 9 logs of killing. If you look at Proteus, which makes a fair bit of urease actually, but nothing like H. pylori, even Proteus after pH 3.5 was killed. And so after 30 minutes or so, pH 2.5 and less is killed. However, if you put Helicobacter into this artificial gastric juice with a little bit of urea present, you can see the Helicobacter is the dark line here. 10 to the 6.5 of the bacteria persisted. And so you've still got 1 million bacteria per mil living in that gastric juice after 30 minutes at pH 1.5. So that's why Helicobacter pylori doesn't care about all those other bacteria. It's the only one that's going to be living in your stomach. So what happens, we published this paper, and you'll see the urea and the hydrogen ion starts attacking the wall of the bacteria. It breaks down the urea to make bicarbonate and ammonia. So even though the environment has pH 2, inside the bacteria it's pH 7, pH 6, something like that, which is pretty normal for bacteria. And so urease protects H. pylori from acid. So what's the use of this? Well, guess what? If you take a biopsy sample, you can do a rapid urease test. So instead of culturing the bacteria in a very fancy research lab with special incubators, you just take a biopsy and you put it into this gel called Christensen's agar, and you'll see that it generates ammonia and the pH changes, and you see a red color, just like litmus paper really. And you see this is probably an early one, and after half an hour you see these red blotches, and that one, that little biopsy there is no urease, so no Helicobacter. And if you make a concentrated form of that, you can see this is a big red spot from a biopsy just after a few minutes, and a colony of Helicobacter is much stronger even than that. So that was the early rapid urease test for Helicobacter pylori, and it became a commercial test called a CLO test, which means Campylobacter-like organism test. And millions of those tests are sold around the world each year. And generic tests are in every country, and at least 30 million tests are done per annum. However, you still needed to have an endoscopy. So the final thing is, how could you diagnose Helicobacter accurately without an endoscopy? Well, hang on a minute. What about this? What about if we take an isotope of urea? And so there's the urea molecule, and on this carbon, we've got a carbon-14 isotope. That's a label, it's a radioactive label, it's just a tiny, tiny safe amount. And you can use another isotope, carbon-13, which is not radioactive if you want to, but really no difference. And you put that in a capsule, so the patient eats the urea, drinks it on a fasting stomach with some water. If the H. pylori is present, you see that that urea is broken down into CO2 and ammonia. The ammonia just goes into your blood harmlessly, and the CO2 goes into your blood and then through your heart comes out in your lungs. And so about 5 minutes after you swallow the capsule, we can detect that isotope of CO2 in your breath, and you can collect it in a special solution, put it into some fancy kind of Geiger counter, and you see these little counts there. If there's lots of counts, usually more than a few hundred, that means you have got Helicobacter. And if you have treatment, we can do that test again. And if it's back to normal, no counts coming out in the breath, you say your Helicobacter is now gone. And that is just as accurate as a pathologist doing a biopsy. So now every doctor in the world almost could easily do this kind of test, and no need to be a gastroenterologist or have endoscopy. So saving everybody thousands of dollars. And so that's the story of how curiosity-driven research led to the Nobel Prize. And here we are, Dr. Warren and I, receiving our Nobel Prizes there at the ceremony in Stockholm in 2005. In the middle, that's my wife, Adrienne. And when you're doing all this hard research, it's a lot of sacrifices for your family. So it's great to have a smart, dedicated person helping you stay in a good mood and succeed with all that hard work that you're doing. I'll just say here that my main affiliations now are with the University of Western Australia, Marshall Lab for Biomedical Engineering, and the Shenzhen University Marshall Lab for Biomedical Engineering, and you'll probably see a few more references to those things in the future. And down here on the right, you'll recognize that this is a QR code for WeChat. So you can all take a photo of that if you like. Send me a red packet. And my WeChat name and my Twitter name is @barjammer. Thank you.
Nice to meet you on air. How are you?
Great. So that turned out to be quite a lecture. I think people liked it because it's kind of like a long YouTube. People can see all the details of it.
So before I come to the questions from the audience, can I just ask you my burning question? My question was, when you drink the bacteria into your stomach, did you ever have a second worry about what would be the consequence, because there was no effective cure at that time of the bacteria?
Did I have any concerns about drinking the bacteria? Well, whenever you do anything for the first time, there's a bit of an unknown. And I say to people, it's like taking your first bungee jump. And actually, I'm never going to do a bungee jump myself, but at least you can look at the science of it, and you can say a lot of people have done it and survived, and the ropes are quite strong, check everything, but you still feel a bit nervous when you do it. So it was like that with Helicobacter. I knew by then that about 40% of people in Fremantle Hospital, just visitors or blood donors, were infected with Helicobacter. And so I knew it was very common. A lot of people had no symptoms. So it wasn't always going to lead to ulcers or cancer. And the other thing is that I had taken the bacteria from a patient who I'd been able to cure using some antibiotics and bismuth. So metronidazole and bismuth were given to this patient, and I knew the organism was sensitive to metronidazole. So I had a bit of information. We'd been working for a few years by then, and I was fairly confident that I could eradicate the bug. Overconfident, in fact, because someone else who tried to repeat the experiment in New Zealand was unable to eradicate the bacteria, and he had stomach trouble then for about 3 years afterwards. So there's always the unknown, and it's bad policy. It's not good science to do experiments on yourself because it's not objective. How could you really be certain that the bug was causing your symptoms? Were you just imagining it or had something else? But we did have one thing that was objective, and that was the biopsy. So we had a biopsy beforehand, and then we had a biopsy in 2 weeks later. And we could see the infection, the inflammation, the white cells, and the gastritis had developed, presumably as a result of the bacteria. So I could start from that point, and then the rest of the data I obtained from that research was softer data, if you like. So it turned out all right. I learned a lot, and we created a hypothesis which turned out to be true. So it was 10 years before people really believed it after that.
There are a couple of people in the audience who ask why not use an animal model, and if animal model doesn't work, is there any other technology like organ on the chips or other technology that will be able to avoid you from drinking the bacteria yourself?
But you asked me about could you do it a better way? Well, now we do have animal models, which is gerbils. Mongolian gerbils are quite a good animal model. And the second thing is you can do studies of organoids, which are cultured gastric glands, if you want to look at things like attachment. And you can even do attachment with sort of GMO, genetically modified cells, removing and adding back the different receptors for Helicobacter. So you can go a long way now in vitro, which is much better than human experiments. So we still do some human experiments. We've got some information from that, but mostly you can do laboratory work. If you have a good plan and a direction, then it's certainly going to be worthwhile doing that.
Okay. Well, just one more question before I pass on to the audience to ask you directly. You mentioned also that despite your findings, your paper was rejected, and nobody believes that a bacteria can survive in the stomach. So, what was your feeling at that time, and what is the right attitude when what you firmly believe in was rejected by the world and your paper is not acceptable? What was your feeling at that time, and how would you advise young people to deal with this?
When the paper was rejected, uh, Warren and I had already been testing our own hypotheses. So when you're in research, you want to try and be objective, and you want to find out why people might not believe your new discovery or new observation. And then you should test your own hypotheses critically, because instead of designing experiments which prove that you are right, you should try to design experiments which prove that you are wrong. It's much easier if it's a false observation or false result. It's much easier to prove something is wrong. It's very hard to prove it's right, and you only succeed with a new paradigm, if you like, um, when you've tried to prove it wrong many times. And this is what the scientific process is. Scientists are going to try and prove that you're wrong, and you should do that yourself before you really start trying to promote your new discovery. You say, yeah, I've tested that, and we found this or that. And so you should try to knock down your own hypothesis several times in the lab before you actually move on to the next step, rather than just coming out with it and saying, I want to get an early publication. Obviously, you can do a very small publication, like a local meeting with a poster and discussing it with your peers. That's very, very important and very useful, and it does fix the timeline of the discovery. So that's important. But you really need to be able to test your own hypothesis quite a lot before you move on. And so I had done that. So we were not particularly worried about all the criticism that we might have and people not believing this idea. And that's why we were trying to do animal experiments as well as human experiments.
But when will you say enough is enough and you will give up? You will not do it anymore?
Well, I notice I don't have as much energy as I used to. And I try, the secret is to find good students, PhD students, postdocs who maybe have got their own research skills that want to work in different angles of it. And then they are doing the hard work nowadays. I've got other activities which I also do. So I'm not working like 6 days a week on the same Helicobacter project. But because of a long history of observations in clinical medicine and in laboratory research related to Helicobacter, I can often see flaws in the experimental technique, or say I know that this has already been tested. So I think you can accelerate the process a little bit if you've got a couple of senior people overlooking things in the lab. So I still have that role. I probably continue to do that for another 5 or 10 years, and then I might say stop.
Okay, there is a lot of questions from the audience. Some are very scientific. Some ask you, how does H. pylori taste? I'm going to ask Lo Yi Choi to ask you some questions, because she actually asked quite a few of them. Well, she actually asked you, well, with so many people infected by H. pylori, but only a small number of them develop ulcers and cancers, why is that so? What contributes this difference in the outcome of the infection?
There's 2 or 3 things. Firstly, to develop an ulcer, you do need a fairly strong acid level, and that has happened more in the 20th century. Prior to that, nutrition is not so good, and often in Asia, the nutrition was not so good, so that people that I tested actually in Brazil were only producing half as much acid as Americans. And so that means that they were unlikely to get ulcers. The low acid state, however, and the severe gastritis that comes from lifelong Helicobacter is a risk factor for stomach cancer. So people that weren't getting ulcers were getting stomach cancer. So they're the things that would be affected by your lifestyle and the quality of your diet. And then the next thing is some Helicobacters are more inflammatory, so that if you have one of those Helicobacters, you're more likely to get more inflammation and more disease. So all those things interact. Luckily, Helicobacter, like a successful bacteria, has 80% of people asymptomatic. They just pass it on. And you don't even know that you have it. So nowadays, it's kind of a screening test. When you have your thyroid function done at age 40 or 50, you might probably have a Helicobacter test of some sort in a lot of countries now. So sorry about that echo. It's fixed, but I realize it was a tricky issue.
Yeah, one last question from Japan, Barry. This is a Japanese doctor, I suppose. He asked you, what is our next battle? Now H. pylori has been eradicated, and there's no more ulcer. And what do you think is the next battle against different type of diseases in the stomach?
So apart from Helicobacter, the main problem is gastroesophageal reflux, I suppose. And so we have got the medications to combat that, the proton pump inhibitors. So we're very lucky these days not to have quite so much untreatable stuff going on in the stomach. And so one issue is, can you predict a person who has got a cancer risk in the stomach? So people looking at things like the microRNAs and different types of histology in the gastric mucosa. So that's underway. That'll be solved probably in the next few years. But in the year of COVID, the second or the third year of COVID, I think we need to look again at what we do with infectious diseases. And we're worried about Omicron, but really all common colds and many of these little viruses that we catch all the time are so infectious, and no one has really tried to solve that problem. We all spend a week being unwell each year with a cold, perhaps. Our productivity goes down. And, we don't think about that like we do with COVID. It's not going to kill anybody, but certainly someone's walking into your office, they're touching your laptop computer, you're picking up the microphone, opening the door. You're going to be picking up viruses all the time. So obviously surveillance for all these different little viruses and rapid production of vaccines is where we're going to go. And at the moment, everything's still having needles. But the future is, I think, that we will develop an oral vaccine. Oral vaccines with a production time of about 3 months, and people can buy them in the supermarket. So that is the holy grail with vaccines. And you take the doctors out of the equation. People can do their own home test. They can take the latest oral vaccine, different flavors, etc. And so one of the angles I've been working on for years is using H. pylori, something about H. pylori to get antigens into the gastric mucosa or into the intestinal mucosa, because you do develop antibodies against Helicobacter and it's in your stomach. So at least your immune system thinks it's a pathogen, tries to react against it. So you might be able to use one of H. pylori's tricks to develop oral vaccines. So that would be the next generation of vaccines after RNA vaccines, I think.
Okay. Professor Marshall, thank you very much for your time and your inspiring talk. I think time is up, and we have to say goodbye here. Hope to see you again very soon. Bye-bye.
Okay, thanks, Joseph.